Hi, my name is Alexa Alvarez. Today is July 23rd, 2020. We are working on Lab 5. And Lab 5 consists of bacterial morphology and staining techniques. Today we're going to be creating direct and negative stains using different stains, and we are going to also be looking at them under a microscope. So I think the easiest way to do this, since it looks like we're going to be creating quite a bit of a couple slides, well, not a lot of slides, but a couple slides. There's a few steps to create each one involving heat and washing or rinsing, and using the candles to kind of cure the cultures on the slides. So I'm going to start with the first one, which we are going to specifically take a swab of, again, like in the prior lab, we're going to take a swab of our tooth and gum line to grab some dental plaque. We have sterile swabs here, so we're going to get working on that. So I have already labeled and created the circles. So the instructions asked us to put one direct and then M. So this is our first slide. It's a direct stain, and we are going to swab the mouth. So that's what the M is for. So on the back side, I did create the circle just so that I know that we're going to focus our sample right in that little circle. So the circle is written on the back side, the letters are written on the front side, and we're going to get going. Okay. Guess I don't know how much is enough. And so I think it's pretty saturated. And then now I'm gonna smear it in the little circle. Okay. Then we are going to turn our candle on. I didn't have a clothespin, so we're gonna improvise on here. So I'm actually gonna use our clamp. There we go. Oops. So now we're gonna put the flame on the underside of the slide, and it says pass the slide over flame 5 times no more than a few seconds. So we don't want to burn this. Okay, now we're gonna let this cool for about 10 minutes. And then we're gonna work on staining the slide, and it looks like we're gonna use crystal violet. So while this dries, I'm gonna turn that off, and I will be back in a few minutes. Also, by the way, before I forget, not very good at this. So this is our card. So the first, the second one shows kind of our assignment for today. So bacterial morphology with staining techniques. Okay, now I'll be back. All right, so we're back. So now we're gonna stain this with our violet stain. So I did end up cleaning the back side of the slide because it had quite a bit of soot in it. So I just grabbed a little Kim wipe and I just wiped it clean to get it as clean as possible, simply because if we're gonna put this under a microscope, we're not gonna be able to see. So I got that cleaned, and now we're gonna go ahead and stain it. So instructions say that we have to put this in a little tray, and we're gonna add a couple drops of the crystal violet, and we have to let it sit for about a minute. Once we do that, we're gonna rinse it, and we have to kind of blot it dry to make sure there isn't any excess liquid, and we should hopefully see some of the cells that will absorb some of the stain. After that, we'll go ahead and take pictures and upload. All right. Okay, so we're at about a minute 20, so we'll be back in 60 seconds. All right, so we have rinsed the slide from the purple with the purple stain. You can kind of see a little bit, I don't know if you'll be able to, but you can kind of see where the stain has kind of resided. So we're just going to go ahead and dry this off. I think you can kind of see up against there. We're going to take as much water off from here. Very carefully. Okay, so I got as much water off of this as possible. Yeah. All right, so that's good to go. So now what we're gonna do is we're gonna take a picture. Once we do that, we're gonna put it under a microscope. We're gonna take a look, find the best magnification to view the cells. We're gonna upload the pictures, and then we're gonna go on to the negative stain. I'll be back. Okay, so we just finished with the direct stain of our gums and tooth line. So this is all done. I don't know if it's a little hard to see, but you can see some of the staining right there. So I was able to find some pretty decent cells in there. So now we're going to create our negative stain. So part of what we have to do with that is we're going to get a clean slide, and we are going to label it. And this one we're going to label 2-neg-M, and so 2 meaning it's going to be the second slide. The neg is for negative stain, and then M is still for our mouth. Okay, so that's done. Now we don't have to circle on this one, so all we're going to do is we are going to open our Congo red stain. That's this guy, and I am going use scissors to cut this. I'm gonna put the scissors on a paper towel. I'll leave that in there. So what we have to do is we have to place a drop of this stain as close as possible to the written side of the slide. Let's see. Yeah, just one drop. Let's see. Okay. I'm gonna put that back in here so it doesn't spill. Now we're gonna grab our second swab and we are going to, vigorously it says, swab our mouth again. So, I think that's good. Get the a lot of times I think we get plaque closer to our molar, so I was trying to get in there. So now what we have to do is we have to place the swab into the Congo red stain. And we're gonna agitate it, and that way we can transfer from the swab to the stain. Let me just do one there. Now we're gonna get a clean slide and we are going to smear the liquid down. So it says to hold it at a 30-degree angle and spread the dye across the slide. So do that. Go. It's a little hard to see. See a little bit. You can see it up here and a little bit down there. Now we have to let this completely dry, it says. So yeah, it looks a little bit wet, so we're gonna leave that here. And we will be back. So we're gonna do another picture of the slide. We're gonna take a picture of, also we're gonna put it under the microscope and we're gonna look at it at all the magnifications to see at which magnification we get a good image of cells. We're gonna record that, and then I think we'll be done with this portion. We'll be back. Okay, so now that we have finished with viewing our negative slide, we are going to start our part 2 of our exercise. So the first 2 slides we worked with, those will go in the trash. We just have to get them sanitized and sterilized, and then we're going to toss them. So now we're going to work on doing, what is this one called? Differential stains. So we are going to work with our inoculated broth. So we have yeast, we have epidermidite, I can't say this, epidermidis, and we have E. coli. So what we're gonna do is a multi-step process of first we're gonna label each slide, and then we are going to put a circle in it at the bottom, very similar to what we did with the first cheek swab, tooth and gum line swab with the crystal violet. And we are going to stain it. Well, first we're gonna heat fix it, and then we're gonna stain it with first crystal violet. We're gonna wait a few seconds, and then we are going to stain it with iodine decolorizer as well as saffronin. And we have to let that completely dry. And once we're done with that, it looks like we're gonna have to take some pictures, and we will have to look at everything under a microscope. So we are gonna go ahead and get started with labeling. And that's that. Okay, so I have to get some alcohol in a cup so I can sterilize my inoculation loops, and then we're gonna get going with getting some of these cultures transferred onto the slides. Be right back. Okay, so we are going to get started with getting tubes sterilized, transferring with inoculation loops, and kind of getting the staining process started. Okay, we have to turn the candle on. First, we have to sterilize this. Doesn't smell very good. Okay. Pregnant noses are very sensitive, so. Okay, so I'm gonna make sure this is really dry. We're gonna transfer onto here. Then we have to sterilize this again. And then we have to close it very quickly. All right. pass this through the flame. And we're gonna let this kind of just sit for a few minutes. So we're gonna do the same thing with the yeast and with the E. coli. Hopefully this doesn't smell bad. So sterilize. It does. This is gross. Oh, this isn't yeast. It's E. coli. Okay, so very carefully. Pass this through the flame. We're going to sterilize this again and seal it. And last, we have yeast. Good. Drying up. Oh. Okay. And then we're gonna re-sterilize this and we're gonna close it. Okay, so now that we have these all ready to go, we are going to clean the soot off of these, and we're gonna go into my restroom and we're gonna do the staining process. Since I need running water, it'd be easier than me going back and forth. So I'm gonna grab a little sheet. Get this cleaned off so we can see our samples in the microscope. Okay. Make sure these are nice and dry. All right, let's go. Okay, so we have our staining tray. We have our, it's kind of hard to see, but I have the three slides there. I'll put them actually on top of here. There we go. So we're gonna start with the epidermidis, and we're gonna put that in here. So what we have to do is we're gonna first stain with violet, then we're gonna add 1 to 2 drops. We're gonna let it sit for about a minute, and then we're gonna rinse it, and then we're gonna stain with iodine, couple drops of that. We're gonna let that sit for another 60 seconds, and then we're gonna rinse it, return it back in here, and then with the decolorizer, we have to wait 30 seconds instead of 60 seconds. Once we do that and we rinse it, then we add saffronin, which we wait another 30 seconds. We have to rinse it, and we have to let them fully dry for a few minutes. I don't know that it'll take too long. So we're gonna get started. That one. And we're at 1:39. I think I'll be back. That way I don't kill some dead airspace, dead air time. All right, so a minute has passed. So now we're gonna put this through a little bit of running water, get the violet stain washed off, and then we're gonna move on to the next 3 stains. Okay, so water is coming out clear. All right. Now we're gonna add some iodine. And 2 to 3 drops. Now we have to let this sit for a minute. Be back. Okay, so a minute has passed. Now we're gonna rinse the iodine off. Yeah. Okay. Now we're gonna add 2 to 3 drops of decolorizer. I'm just gonna wait 30 seconds on this one. just gonna keep track up there. Okay. Like that. And last but not least, saffronin. And this one, we just wait 30 seconds as well, 2 to 3 drops. Okay. Got that. And we're going to do the same thing with the next 3 slides. I'm sorry, 2 slides. So now we have E. coli. Decolorizer. Okay, I'll be back after a minute. Okay, a minute is up on the iodine. I'm gonna rinse. Okay. Okay, now we're gonna add decolorizer, a few drops, and then wait 30 seconds. Perfect. Okay. Last but not least, saffron. Wait another 30 seconds. Perfect. Okay, that one's done. And last but not least, yeast. Okay, be back in a minute. Okay, our minute is up. Now we're gonna rinse. Next is iodine. Wait a minute, be back. Okay, that minute is up. Okay. Up next, decolorizer. 30 seconds. Okay. Lastly, saffron. 30 seconds. we're done with that. Now we're just gonna let these sit and dry off completely. Get this mess cleaned up because there's dye everywhere. And I'll be back. Okay, so these have pretty much dried. There may be like a couple drops of water, which I'm gonna let them sit for just about a minute or two. And once we do this, then we are going to put them under the microscope and take a look and see if we can distinguish the different cells with the different coloring stains and take some pictures, and I think we will be done with this experiment. All right, talk to you guys later.